Hello everyone. Today we are going to solve this GATE question from 2023 Life Sciences. Question number 103. The question states that the length of a double helical DNA molecule is 13.6 kilometer. If the DNA double helix weighs 1 into 10 to the power minus 18 grams per 1000 nucleotide pairs, and rise per base pair is 3.4 angstrom, then the weight of the double helical DNA molecule in nanogram will be. So we have to calculate the weight of the double helical DNA molecule in nanogram unit. And we have to put the integer value here. So this is a numerical answer type question or a NAT. Now before solving this question, just for some background, we will discuss the structure of the DNA. Now we know that nucleic acids, there are two types of nucleic acids, DNA and RNA. And DNA, which is a double-stranded molecule, this DNA exists in a double helical form. Now this double helix model was first described by Watson and Crick. And we also know that DNA is a polymer of nucleotides. Now what are nucleotides? So this is the structure of a nucleotide. And a nucleotide is composed of three components: one nitrogenous base, one pentose sugar, and one phosphate moiety. So these three components together form a nucleotide, and when a chain of nucleotides are joined by phosphodiester bond, that forms a polymer which is called the DNA. Now there are five types of nitrogenous bases: adenine, thymine, guanine, cytosine, and also uracil, which is present in RNA. But in DNA, these four nitrogenous bases are present: uh, adenine, thymine, guanine, and cytosine. And we know that these bases, they also form hydrogen bonds among each other. So adenine forms two hydrogen bonds with thymine, and cytosine forms three hydrogen bond with guanine. So this hydrogen bond formation creates these base pairs, which is the fundamental reason why the two strands of DNA exist in a double helix format. So because of this base pairing of the nucleotides, that is the base pairing between the purines and the pyrimidines. So due to this reason, the two strands of DNA, they are complementary to each other and they form these base pair interactions. Now these base pairs are projected inwards, and the backbone of this DNA, this particular backbone, is formed by the sugar and the phosphate. So the sugar and the phosphate, they form the backbone, and the nitrogenous base that is projected inwardly from the sugar phosphate backbone. And these nitrogenous base, they form base pairs with particular nucleotides. So adenine forms base pair with thymine, and guanine forms base pair with cytosine. Now, this double helix model of the DNA, so as it is also mentioned in the question, that the rise per base pair is 3.4 angstrom or 0.34 nanometer. So this is the standard B form of DNA. And in the standard B form of DNA, we can see that in this double helix, the rise between base pairs is 3.4 angstrom, which means the distance between two base pairs, this particular distance is 3.4 angstrom. And the rise per turn is 3.4 nanometer or 34 angstrom. And we know that there are approximately 10 base pairs in a turn. So, 3.4 into 10 also gives us 34 angstrom. So we know that in a uh, turn, in one complete turn, there are approximately 10 base pairs. So this is the information that we need to solve the question, which is also given in the question. So now let's get back to the question. So what is given in the question is that the length of the DNA, which is 13.6 kilometers. Now here we are considering units in of angstrom while studying the helical structure of the DNA. So what we will do is, this is given in the question, we will convert the kilometer to angstrom. So 13.6 kilometer is 13.6 into 10 to the power 3 meters, and we know that 1 meter is 10 to the power 10 angstrom. So we will multiply this with 10 to the power 10, and the answer that we get is 13.6 into 10 to the power 13 angstrom. So that is the length of the total DNA, which is given. Now it is also given the rise between base pairs is 3.4 angstrom. So therefore, from this information, we can calculate the number of base pairs present in this length of the DNA, because the distance between two base pairs is 3.4 angstrom. So we can, from this, we can calculate the number of base pairs. And how do we calculate the number of base pairs? We simply divide it. So divide the length by the rise between each base pair. So 13.6 into 10 to the power 13 divided by 3.4, which gives us the number of base pairs as 4 into 10 to the power 13. So there are 4 into 10 to the power 13 base pairs or nucleotide pairs present in this particular DNA molecule. Now let's come to the weight of the DNA molecule, which is given here in the question, that 1 into 10 to the power minus 18 grams of weight per 1000 nucleotide pairs or per 1000 base pairs. So 10 to the power 1000 means 10 to the power 3. So 10 to the power 3 base pairs weigh 1 into 10 to the power minus 18 gram. 1 into 10 to the power minus 18 gram per 10 to the power 3 base pairs. So now one base pair will weigh 1 into 10 to the power minus 18 by 10 to the power 3. So simply by following unitary method, and we have to calculate the number of base pairs that we have got here. So therefore, this number of base pairs, 4 into 10 to the power 13, how much will it weigh? So 4 into 13 base, 10 to the power 13 base pairs will weigh 1 into 10 to the power minus 18 into 4 into 10 to the power 13 divided by 10 to the power 3. So therefore, we will get this value 4 into 10 to the power minus 8. Now, if you look at the question is asking the weight of the DNA molecule not in grams but in nanograms. So the unit, we have to be very careful while answering these questions. We have to be very careful of the unit. Otherwise, we will get a wrong answer, because we do not have to put the unit here along with the answer. We will just have to put the integer value. So we will only have to put the value without the unit. So the unit has to be accurate in the unit in which we calculate. So now we have got this answer in grams, 4 into 10 to the power minus 8 grams. That is what is asked, the weight of the DNA, but in nanogram units. So what we will do is, we know that 1 grams is equal to 10 to the power 9 nanograms. So we will multiply this value with 10 to the power 9. So 4 into 10 to the power minus 8 into 10 to the power 9, which gives us the value 40 nanograms. So that is our answer. So if we go back to the question, the weight of the double helical DNA molecule in nanogram will be 40. And that is your answer.
For more information, please follow this NPTEL course Biochemistry. Thank you.